Hi, I'm Richard Sever from Cold Spring Harbor Lab and BioArchive. With me I have Steve Henikoff from the Fred Hutchinson Cancer Center and Howard Hughes Medical Institute. Steve, welcome to Cold Spring Harbor.
Thank you, Richard.
So we're here at the annual symposium, and you gave the opening talk last night, which you introduced a veritable alphabet soup of centromere-associated proteins. But what I was, and we must get into that, but I was very interested that you started off your talk with a slide from about 145 years ago. So was that the first description? Tell us a little bit about that.
Those were drawings from Walter Fleming. Now, Walter Fleming had introduced the term chromatin, and that was back he published that in 1882. So chromatin actually precedes the rediscovery of Mendel's laws, precedes the beginning of genetics. And what he called chromatin was actually what I showed, his drawings, which showed chromosome segregation I think it was the newt. And he showed these beautiful drawings of chromosomes that were being pulled to the poles at mitosis. And what was particularly striking to me was that you could see everything. You saw the centrosome, you saw the spindle fibers that were holding on to the chromosomes. And what he was very careful to show was for each of the centromeres, each of the connections between the spindle fiber and the chromosome, he showed a little darkening there. So he actually saw centromeres, or heterochromatin as we call it now, pericentric regions, whatever. He saw them and he colored them in. So actually centromeres were discovered before before the chromosome theory of heredity. But this is what the cytologists were showing us. So then with the rediscovery of Mendel's laws, people knew about chromosomes and they kind of put it together, and that's where the chromosomal theory of heredity came. But it's interesting that what was so compelling was seeing the chromosomes separate on the spindle pole. So I thought that, well, centromeres were really the first genetic loci after Mendel's work.
Right. And what's interesting is sort of then fast forward 100 years, and by that stage, the 1980s, we knew a lot more about centromeres. But what's kind of interesting is this is a sort of fundamental, the first genetic locus identified. It's this fundamental aspect, but so different in so many different species. One of the points you made.
So that was one of the points. So let me just go through that just a little bit. The beginning of the molecular study of centromeres was this paper that I pointed out from Louise Clark and John Carbon, where they actually showed that if you just take a little piece of DNA that comes from the genetically mapped centromere, you put it on a plasmid, so you make a chromosome that will segregate normally during both mitosis and meiosis. So that proved that piece of DNA was the centromere. And I remember when that paper came out, it was quite revolutionary in a sense, and I thought, oh, they're all going to be that way. So we were studying heterochromatin in Drosophila at the time, and I thought, oh, it would be really very simple because it's just going to be something in there among all those repeated sequences that we can't make any sense out of. There's going to be some little magic sequence in there, and that was not to be. So we ended up, so a lot of progress was made in studying the budding yeast centromere, and even fission yeast turned out to be more different. And then it turned out that most organisms, nearly all multicellular organisms, have centromeres that are highly repetitive, tandem repetitive sequences. And I can tell you, having lived through it, that there was a lot of skepticism at the time that alpha satellite, for example, which turns out to be what our centromeres are composed of, there's some skepticism that there's just going to be homogeneous alpha satellite repeats. Other people thought it might be other satellites and all, until there was the experiment by Hunt Willard and his colleagues showing that you can actually make artificial centromeres. So do the equivalent of the yeast experiment, but you can do it with human centromeres, but you need long arrays of these alpha satellite sequences. So that was a problem. And then there's the fact that, in 2000 or 2001, the draft human genome was published, it's still a gap at all of our centromeres. We haven't assembled it yet because it's so homogeneous, you can't really do much with it. So it's been difficult to study. A lot of progress has been made, particularly in yeast, and understanding centromeres. That's our best example.
So how long? So in yeast, how long is that sequence? This dedicated sequence in budding yeast?
It's 120 base pairs. It varies a little bit. It's between chromosomes. There's 16 chromosomes, and they have canonical sequences that comprise the first element, which is about 8 base pairs long, and that's binding site for a transcription factor. And then on the other end, there is the binding site for a kinetochore specific complex that recruits the centromeric nucleosome, which sits in the middle, but there's only about 80 base pairs for that.
Right.
And so people have been wondering, well, how do you get a nucleosome with 80 base pairs? And this has been a bit of a controversy. I showed some evidence on that. I think it's pretty well established that that it's a smaller particles. There's just less. There are only 4 histones in there, probably.
Right.
Because that's all that you can really wrap 80 base pairs around.
Uh-huh.
And so I don't think that's been so much of a problem. Where it's really turned out to be a problem are the satellite centromeres, the ones in us, plants, and most higher eukaryotes.
So when you say satellite centromeres, this is alpha satellite sequences. What exactly are those?
Alpha satellite is 170 or so base pair unit that gets repeated multiple times, almost homogeneous, but importantly, they're these higher order repeats. And in our genome, for example, the large majority of the alpha satellites for most of our chromosomes is a fundamental dimeric unit, in that you have one alpha satellite unit here and then another. They might only be about 60% or so identical one to the next, so they're really very different. And then you go to the next one, and there's and so that dimer gets repeated, etc.
So that pairs in the same orientation.
In the same orientation, right, and it's just homogeneous. And then, so it's very homogeneous, but then sometimes there's enough divergence that you'll see that there are higher orders above that. So you get even numbered, you get there's dimers, there's tetramers, there are octamers, etc. So the usually even numbered ones for most of the chromosomes. But then 3 of our chromosomes have an odd number. 5 is the basic number. So it gets very complicated from chromosome to chromosome. So that's been one of the problems. But even today, there was a lot of description in the meiosis session about mouse centromeres, which are much simpler in a sense. And they only have 2 different satellite repeats. One of them, the minor, has 120 base pair repeat unit, and the major has a 234 base pair repeat unit, and they don't see higher order structures. So the higher order structures are something that you find, for example, in primates, but you don't find it in some others. We've been looking a lot at Arabidopsis, so the plants have the same kind of structure. They have 178 base pair repeat unit. They don't have higher order structures. So I can go through all these examples, but the one thing that's in common is that there are these highly repetitive sequences that are there, and of course that makes an interesting challenge in order to study them. But it also means it also asks the question why, and
right.
why is it like that?
And do you have any? I mean, given that given this is such a fundamental thing that's shared by anything that needs to segregate its sister chromatids, which is everything. Why it's also different.
Well, so we have a hypothesis on it. It was actually discussed this morning in the session on meiosis, which is, I didn't get into this, but it's what we refer to as centromere meiotic drive. So I'll just explain it briefly. I don't know if there'll be interviews with other people who were working on this, but the way centromere meiotic drive works, it only occurs in female meiosis for the following reason. So female meiosis is asymmetric, and the 4 products of meiosis, only one of them will get chosen to go to the next generation. Because it's asymmetric, there's the egg pole, and then there's the pole which will give you polar bodies. So in other words, only one of the 4 products will move to the egg pole. And it turns out that there is a reorientation process because there's a competition between centromeres centromere from the maternal side and centromere
So
from
we're
the
back to
paternal
selfish
side.
genes.
So it's actually selfish. So we'd actually proposed this quite a long time ago, that centromere drive is really a process in which there's a reorientation. So there's a competition between the maternal and the paternal side, and because of that competition, centromeres are competing to make it into the egg pole. So they're So they're the most selfish elements that can be. They're just repeat sequences, and so the bigger centromeres in some organisms, the bigger centromeres make it to the egg pole.
Oh, I see.
So the stronger centromeres, and that actually was shown very nicely today in talks from Ben Black and from Mike Lamson. They actually showed that this actually occurs in mouse, and they worked out a lot of the details of the process.
Oh, I see.
So we think the why is that centromeres are competing and they're rapidly evolving because of it. It's a Darwinian process of cheating in female meiosis.
Right. Oh, I see. So I mean, it's interesting then because in some respects, conceptually, the next level, which is all this alphabet soup of proteins coming on, it's probably more conserved. Is that
no?
fair to
Actually, so it turns out it's an arms race.
Uh-huh.
So the way we got onto it, Harmeet Malik, when he was a postdoc in the lab, had discovered that what I talked about, SEMP-A, centromere protein A, which is the centromeric histone, which is absolutely essential for
this is the thing that replaces the regular histone H3.
Yes, right. So the H3 variant, we were working in Drosophila, but it turns out to be more general. The H3 variant itself was showing rapid evolution. And we can show what Harmeet discovered was if you look at the excess of replacement over synonymous changes, you see a large excess of replacement over synonymous changes. So so we know about arms races where this kind can occur, for example, between a pathogen and host immune system and say a virus that its coat protein will mutate to evade immune surveillance, and then the host immune system will clobber it, and then so you get this arms race going, and it builds up changes on the surface of the viral coat protein. We see the same kind of process must have been occurring for the centromeric histone. But how could that be? Because if it's because that's going to be deleterious, and you'll be dead.
Right.
Because every cell division is going to die. So finding centromeric proteins, and we found that for SEMP-A, for SEMP-C, for basic centromeric proteins are undergoing an arms race. And then we realized, well, yeah, what's the arms race? And then that's where it came up. Oh, maybe it's female meiosis. That's the only time.
Oh, I see.
The only time that you actually can have an arms race.
Right.
And so that's what we think. So we call that the centromere paradox. Why is it that centromeric sequences are evolving so rapidly, both in the size of the centromeres? Your Y chromosome and mine might differ by an order of magnitude in size. So they differ tremendously, and yet they're doing the most basic process we know of in genetics, which is chromosome segregation, which is very, very stable. So how can that be? And we think it's an arms race of female meiosis. And I think there's that was like 15 or so years ago, and I think it's holding up pretty well.
Oh, that's cool. And you mentioned SEMP-A. I think you mentioned in your talk SEMP-A, B, C, T, S, W, X, Z. So this is like a giant protein complexes on top of the DNA that then link to the kinetochore that attaches to microtubules. Is that correct?
That's right.
Yeah.
That's right. So it's on the DNA. Of course, the DNA is wrapping around the nucleosome part. It's also wrapping around its MTWSX. So I mentioned a complex of histone fold proteins that the DNA wraps around, and that's what's important about that is there are two connections to the outer kinetochore.
Uh-huh.
And there's been some question as to where those connections come from the chromatin level. So the two connections is one is SEMT and one is SEMC. That's where we get the alphabet soup. But SEMT has partners W, S, and X. SEMP-A has partners H4, H2A, and H2B. Uh-huh. So if you think about it like that, you've got two parts that connect. And what we show is that they're all actually part of one coherent complex. And we showed this in a couple of different ways, including a new method that I described that we call cut and run, where...
So tell us about this, because I mean, I think up until last night, I think everybody was familiar with sort of ChIP-seq, where you bind your antibody onto the protein of interest, then you sonicate away all the DNA, and this seems like a much cleaner way to get a better signal. How does that work?
The way it works is that we take cells or permeabilized cells or nuclei. So we're starting out with intact cells as opposed to breaking everything up like you do for ChIP, and everything stays intact. We then permeabilize them in such a way that we can get antibodies to go in and find their targets in the chromatin, let's say for a transcription factor or a centromere protein. And then we add a fusion between protein A and micrococcal nuclease. This is all a method that was introduced in 2004 by Uli Lemley. However, it really hadn't been used since till we started working on this and improving the protocol. Protein A will bind to the immunoglobulin G of the antibody, so therefore you're tethering this nuclease, micrococcal nuclease, which is our standard tool for looking at chromatin. And once it's tethered, it has some very nice properties. So what we can do is we can put it at a low temperature so it'll just stay in place during the reaction. We can then add, I should mention, micrococcal nuclease requires calcium. So it's not going to do anything until you add the calcium. You add the calcium, let it go for a few seconds or an hour or something, it doesn't seem to matter. It will cut out on both sides the particle, and then it'll float out through the nuclear pores, and we just take the supernatant and extract the DNA, and it's very simple. And you can do it, and our current protocol takes about half a day. It could even take just a couple hours, and you can go grab a cup of coffee and or you can do it before lunch.
So, but you do it before lunch, and you get a much cleaner signal.
Yeah, as I showed, so being clean is important, because when you're looking at something like a centromere where there's only one per chromosome, or transcription factors, that you have one here that has a footprint of about 20 base pairs, another here, 20 base pairs, and you've got 10 kilobases in between. Basically, when you chew up the whole DNA, like you do with ChIP, you're going to have a certain probability of every little piece of DNA getting sequenced, and that's going to give you a low background. It might be a low background, but you have to do a lot of sequencing in order to see those peaks. With cut and run, because we leave the vast majority of the DNA behind on the beads, and only what floats out that gets released is what you sequence, the background really goes way down, such that we only have to sequence about a tenth as deeply. And because the antibody is not looking at the whole cell contents that get ground up when you do ChIP, but rather it's seeing the intact cells, it's seeing in the nucleus, it's only seeing the surfaces, and that means that the antibody binding is usually very efficient. So we can get down to just I showed where for a histone modification we can get down to 100 cells.
Wow.
So it should be good for low cell numbers. It's all new, but a lot of people are trying it, and I've been getting good reports. So I think it's going to catch on.
Well, that's great to hear. And it sounds. You also said that as a consequence of this work, you'd surfaced more about the centromere binding proteins.
That's right. Yeah. Yeah. So one of the issues that's made study of the centromeric nucleosome controversial, frankly, is in large part because the ChIP method is sort of destructive, because at some point before you add the antibody, you solubilize everything and you can lose some parts. Mm-hmm. That's what we're finding. No matter how you solubilize it, whether you chew everything up with micrococcal nuclease or grind it up, it's going to cause some damage. But because cut and run, the antibody's added when everything is intact. It's added, in fact, immediately at the time that you permeabilize a cell. So they were alive, and then, we get them as quickly as possible, and everything is intact. Because of that, I think we're getting a more accurate picture. So what we see is quite compatible with what we've seen with ChIP, but I think it shows us that what we're looking at was sort of eroded particles, but actually the intact particles are much larger than what we thought. Typically over 180 base pairs over this dimeric repeat unit of 340. And so because they're larger, they can accommodate a lot of the alphabet soup of proteins that we see there, including the ones I just talked about, SEMS A, B, C, and T. We can see that they're all there.
Okay, well, that's great. Well, it sounds like, 140 years on, you're finally getting to the crux of what it is that defines the mammalian centromere. It's been fascinating talking to you, Steve. Thanks very much.
Thanks, Sergey.